Hi, Miss Nikki here. Welcome to chapter 28. We're going to do kind of an overview before we talk about the male or the female reproductive systems. So this one's a lot of definitions, just making sure that we're all kind of on the same page. So this first image, I just wanted to show you, this is an onion root tip and it's going through the phases of mitosis. So we're going to look at the differences between mitosis, which is producing an identical copy, to meiosis, which is producing genetically different daughter cells. I just wanted to make sure that we're familiar with the word perineum. So perineum is bordered by, up on the top here, superior portion. This is the pubic symphysis. Down here we have the coccyx bone, and on the sides here we have the ischial tuberosities of our coxal bones. So you can see the female genitalia and the male lie within this perineum. This is not the same as peritoneum, right? The visceral and parietal serous membrane layers that line the abdominal cavity. Some of the common terms that we need to know, like gonads, right? Gonads are the reproductive organs, ovaries in females, testes in males. And then hopefully we know the gonads produce gametes. So in the female it would be egg, and in the male it would be sperm. These are the sex cells. They're going to unite, and we call this fertilization, and this is how we make new individuals, right? We're also going to talk about lots of different sex hormones. Hopefully we remember estrogen and progesterone for females and testosterone for males. We're also going to come back and talk about follicle stimulating hormone, luteinizing hormone, gonadotropin releasing hormone. Both sexes are also going to have accessory reproductive organs. So we're going to have certain ducts that are going to help carry the egg and sperm to their destination. We're also going to have lots of glands that are going to help with secretion. So we'll move into those as we go through the slides. So what is sex? Sexual union. You can call it copulation, you can call it coitus, you can call it sexual intercourse. Fertilization occurs. The female is what's providing the support, the protection, the nourishment. So the male is providing basically the DNA. So we're going to see a flagellum, a tail. We're going to see the sperm have this mid piece that has some mitochondria. We're going to have DNA, and then they're going to have some enzymes in the cap part. So basically the sperm are dumping their DNA inside of the female sex cell, which would be the egg. And then the egg and the sperm are going to unite if fertilization occurs, and then the egg is going to provide the support, the female reproductive tract, protection, nourishment, because the fertilized egg or zygote has to implant in the uterus for development. I have the term puberty highlighted. So what is puberty? It's sexual maturation, males and females. When you start seeing external sexual characteristics develop, so breast enlargement, pubic hair growth, this is when the gametes begin to mature, the egg and sperm. And then we're going to see the secretion of sex hormones. So the majority of sex hormone secretion will come from the testes in the male or the ovary in the female. Please remember you still have your adrenal cortex supplying smaller amounts of all of these hormones. So now we have to go back and talk about gonadotropin releasing hormone, follicle stimulating, and luteinizing hormone. Hopefully we know that GnRH is released from the hypothalamus, and that triggers the anterior pituitary to release follicle stimulating hormone and luteinizing hormone. So this is when we're going to see the ovaries and the testes start producing more sex hormones, and this starts the process of making eggs and sperm, and then starts the presentation of those secondary sexual characteristics. So gametes, we're going to call the egg, its true name is oocyte. You'll still hear me say egg and sperm, but its technical name is oocyte. Males are going to produce lots of sperm, 100 million a day, right? They're stored. If they're not expelled from the body via ejaculation, they're going to be reabsorbed. Gametogenesis is this overall umbrella. So this is the process of forming human sex cells. So the whole thing, whether we're talking males or females, right? So female gametes, the oocyte, the male gametes, the sperm. So gametogenesis, hopefully we can see to make gametes. We're going to see that meiosis is this 2 division. We're going to have 2 separate cell division processes, and they're going to help us create these oocytes and sperm. Before we do that, we have to talk about chromosomes. So we have 23 pairs of chromosomes. So in humans, since this is human anatomy and physiology, we have 23 pairs. So 23 times 2. So we have a total of 46 chromosomes. 22 of them are autosomes, or 22 pairs are autosomes. Autosomes are going to code for things like eye color, hair color, height, skin, pigmentation. And then you're going to have 1 pair of sex chromosomes, and those would be either XY, XX, or XY, and there are some other variations. Here we can see female, male. So these are the genetic chromosomes, the sex chromosomes, the genetic coding for male or female. 1 member of the chromosome pair is inherited from each parent. So what they're saying is, if this is you, this is an adult cell, right? So this is, I'm just going to draw 1 set of chromosomes. This is me. I'm going to say this is a cell that's in my ovary. Where did this chromosome come from? It came from my mom. And where did this chromosome come from? It came from my dad. So right now I have these 2 chromosomes. This cell is in my ovary, and this cell is going to go through meiosis. The term diploid is if you have 2 of everything. So 23 pairs, that's 2 of every copy. That's a total of 46. Haploid is 1 of every chromosome. So this is 23 chromosomes total. Meiosis starts with a diploid parent cell, and it produces haploid daughter cells called gametes. So when you see gametes, you're thinking egg or oocyte and sperm. So they're saying you start with a cell that's a diploid, 2 of every chromosome, 23 pairs, so 46 total. You start with a diploid parent cell, and you are going to produce 4 haploid daughter cells. The diploid parent cell in the gonad, the ovary or the testes, 1 set of chromosomes came from your mother and 1 came from your father. We're going to take this cell and we're going to create either egg or sperm. Mitosis is somatic cell division. Usually when we see somatic, we think of body, right? Whole. It produces 2 daughter cells, and they are genetically identical. There's no crossing over. We start with a diploid cell, we end up with a diploid cell. This is the majority of the cells in your body. They undergo mitosis. So imagine if you cut yourself and you need to grow new skin cells, you're going to do mitosis from the existing skin cells that you have. Meiosis is called cell sex division. You're going to start with a diploid, and you're going to end up with 4 daughter cells, and they are all haploid. So basically, you're having the number of chromosomes. And we're going to see some crossing over happen between chromosomes where pieces of DNA, pieces of genetic material are exchanged. So the 4 cells all end up being genetically different from each other. We're going to see cell phase prior to meiosis is called interphase. This is when the DNA is going to be replicated. So you're going to take this cell that you started with, right? And it's got 46 chromosomes, right? 46 total. It's got 23 pairs. So this is chromosome 1 and this is chromosome 1, and I've got a whole bunch more, right? I'm not going to draw them all. And you're going to replicate these. You're going to copy them. So now you're going to end up with a cell where this one copied itself and this one copied itself. So these replicated chromosomes now consist of 2 sister chromatids. So this is a sister chromatid and this is a sister chromatid. So copies, right? Structures with identical copy. They're attached at the centromere. Sister chromatids are not the same as pairs of chromosomes. So these are pairs of chromosomes. This is a sister chromatid. It's a replicated copy, right? It's a copy of this chromosome. Meiosis 1 begins after interphase. This is when homologous pairs of chromosomes separate. So what did we do? This is what we started with, right? We started with this diploid chromosome, right? 2 of everything. Then we said we copied. So now we have this and this and this. So I'm going to put an X here so we know that these 2 are copies, right? And then this one has an O. I'll put an O there so we know that these 2 are copies. So these are sister chromatids. These are sister chromatids. All 4 of these are homologous pairs or homologous chromosomes. So sister chromatid, homologous chromosomes. So each with 23 chromosomes replicated, sister chromatids are held together at the centromere. And then we're going to go through the 4 phases of meiosis 1. So this is a great image to see or walk through meiosis 1 where you can see the crossing over. So here are the sister chromatids. These 2, these are sister chromatids. These are sister chromatids. All 4 of these are homologous chromosomes, right? Where did you get the original copy from? You got them from your mom and dad, and they're now you, right? This was your cell. You had 1 from your mom, 1 from your dad, and all we did was copy them, right? But in the copying process, well, I should say it's after, it's in prophase 1. We copy them first, and then we're going to cross over. So the crossing over occurs between homologous chromosomes, where you can see pieces of genetic information were swapped. So you can see that this chromosome is mostly dad, but it swapped a little piece of mom. And now you can see over here, there's a little piece of dad. Then the chromosomes line up on this metaphase plate, and they get pulled to opposite poles. That's anaphase. And then we see telophase. We're going to put the nuclear envelope back around the chromosomes. And in here, we have 2 of every chromosome, but it's mostly material from dad or mostly material from mom. So you're going to see them talk about these cells as being haploid. Haploid means half of the genetic information. So this was chromosome 1 that got copied, right? So we start over here. We had chromosome 1. We copied it. And this one over here is this chromosome that we copied. So what they're saying is this cell, for chromosome 1, it only has half of what it started with. It has a duplicated half, but it still only has half of the information, half of the genetic information. So if I said, in terms of genetic information, these cells are haploid. In terms of total number of chromosomes, there are 2 copies of each one. In the second round of meiosis, the second division, we're going to see prophase 2, metaphase 2, anaphase 2. Now the sister chromatids line up and separate. So you can see we're dividing through that centromere that was holding them together. We're pulling them to opposite poles. And we're going to end up with 4 cells that are going to have 23 chromosomes each. These are haploid. They have half the genetic information, but they also have recombinant. They also have mixing of chromosomes. So we now have 23 chromosomes, but each of these daughter cells is genetically different from the next. This is how you can have 4 brothers and sisters and they all look different. Maybe mom has blonde hair and blue eyes and dad has brown hair and brown eyes. You can have one kid that looks exactly like mom, blonde hair, blue eyes. You can have one child that looks just like dad, brown and brown. You can also have mixed, where they might have dad's brown eyes, but they have mom's blonde hair. Or you can have the opposite, where they have mom's blue eyes, but they have dad's brown hair. So the 4 cells that are genetically different from each other. So hopefully we're okay with, we started with 46, then we duplicated all the chromosomes, then we crossed over, then we split into 2 cells, and then those 2 cells split again in this second round of division, and we end up with 4 haploid daughter cells. These are going to become the egg and sperm, depending on which organ we're talking about. There are some issues that can happen. So remember, homologous chromosomes are when we have these guys, right? 4 homologous chromosomes. And you can have a failure of separation. So in meiosis 1, the homologous chromosomes separate. In meiosis 2, the sister chromatids separate. So depending on where there's an issue with division, it can result in a different number of chromosomes. You can have 1 extra, or you can have 1 less than you need. Most common trisomy is trisomy 21, which causes Down syndrome. So all of your other chromosomes are fine. All you have is 1 extra. You're going to have 3 chromosome 21s. And it can cause all sorts of disabilities, right? Heart defects, poor muscle tone, short stature. So the chances as we get older, the chances of having non-disjunction events increases. Difference between genetic and phenotypic. So genetic is genotype. Phenotype, physical. So what do they look like? Physical appearance. So a genetic female is XX, a genetic male is XY. Ovaries are female external genitalia. Testes are male external genitalia. This is a physical attribute. This is what you look like. In males, remember males are XY, on the Y chromosome, there is this SRY gene. This gene produces a protein. Remember, DNA, RNA, codes for protein, right? So that gene, if it's expressed, produces proteins that stimulate androgens. Androgens initiate male phenotypic development. So think testosterone. If the Y chromosome is absent, or if something's wrong with the SRY gene, you will develop into a female physically. You'll still be XY genetically, so genotype, but phenotypically you will look like a female. So female development, there's no SRY protein because there's no SRY gene, and you're going to see the development of female structures like the uterus and the vagina and the uterine tube. At about week 5 or 6, you're going to have, if you're a male, you'll have the SRY gene that's going to produce the protein, and you're going to see an increase in testosterone. I'm going to abbreviate. When that happens, you're going to see this body plan start turning into the male body plan, where you're going to have seminal vesicles that produce semen, right? Seminal vesicles. Still going to have a bladder. Females have a bladder as well, but you're going to see the ductus deferens. You're going to see the testes development, the epididymis. All of these structures we'll talk about at some point. And eventually you'll have the urethra developing as well. In the female, if there's no SRY gene, there's no testosterone being produced, then you're going to have the development of the ovaries. You're going to have the development of the uterine tube, of the uterus, of the vagina. But can you see how here at week 10 to 12, the structures look pretty similar? They're just starting to differentiate. This is a little bit later, right? This is at birth. So, 38 weeks or so later, much more developed. And we'll talk about how some of these structures are homologous, meaning similar, right? So ovaries and testes, right? Uterine tube versus ductus deferens, used to be called vas deferens. So we'll talk about some of these homologous structures in the male and female as we move through male and female reproduction. So are there times that you could have a discrepancy between genotype and external genitalia? So this would be phenotype, right? So yes, it is true. These are intersex individuals. They could have both ovarian and testicular structures, so kind of a mix of both male and female phenotype, right? They can have ambiguous, meaning not clear, right? External genitalia, and genetically they could be male or they could be female. So it's kind of like the genetic inside doesn't match the physical or external phenotype sex. There are other instances. You could be 46 XY, so you're genetically a male, but your external genitalia resembles a female. So your phenotype is female. If you are not producing enough testosterone, remember that SRY gene is going to make testosterone. It's going to make the SRY protein, which is going to trigger androgens to become testosterone. So what if there's some break in this plan, right? From gene to protein. We also have something to do with receptors, right? You have to have receptors on the target cells, all the things we learned back in endocrine. So is it possible for you to be genetically a male, you're XY, but the signaling of events didn't happen, and in utero, your structures turned into female structures because testosterone wasn't present? Yes. For 46XX intersects, these are genetic females, but their external genitalia resembles a male. So the clitoris can be enlarged. The clitoris and the penis are one of those homologous structures. They're made of the same material. Labia and scrotum, same base plan to create either one of those. So also if the fetus is exposed to excessive amounts of testosterone. So you're genetically a female, there's no SRY gene, but you're exposed to androgens and testosterone, and that causes enlargement of the clitoris because it's made of the same material, has the same receptors as a penis would. This could be something to do with the adrenal gland, right? So they just want to make sure they're bringing back in here that pre-puberty, what is producing your hormones? Your adrenal cortex. Remember those? Gonatocorticoids. So definition of puberty, adolescence where reproductive organs become fully functional, right? External sexual characteristics become more prominent, breast enlargement, pubic hair growth. The timing of this can be affected by genetics, environmental factors, health. I think for females, you'll see 8 to 12. This used to be a little bit higher, right? 14, 16. And then I think for males, they have, I think it's 10 to 14, 12 to 14. I'd have to double check that. So the initiation of puberty is going to start with the hypothalamus releasing GnRH, with FSH and LH being released from the anterior pituitary, and then these are going to go to the gonads, either the ovary or the testes, and cause them to secrete sex hormones. Early signs of puberty, you can see breast bud development, pubic and axillary hair in boys and girls. First menstruation, menarche about 2 years after the first sign of puberty. Boys, you're going to see testicle and penis growth. And then this structural changes in the larynx. So more to do with the vocal cords, right? So that deepening of the voice. Females do mature a little bit faster than boys in terms of the timing of puberty. Here are those ages that I was trying to recall. And then sometimes we can have, without a known cause, early development, So it could be brain injury. This would have something to do with the gonadotropin releasing hormone, right? Pituitary, this would have something to do with increased FSH and LH, or there could be some sort of tumor in the gonad, the ovary, or the testes. And this would be increasing estrogen, progesterone, or testosterone, which would start the earlier timing of puberty. So this was just a quick overview, talking a little bit about kind of topics that cover both male and female. Next lecture will be all about the male, and then we'll finally get into the female, and then end the unit with talking a little bit more about development. I'll talk to you soon. Bye.